So welcome back. While you were away on break, I corrected my spelling error. That's one of the advantages of taking a break. So let's take a look at the concept check question. You decide to breed two pure breeding dogs. So basically, these are two dogs similar to the pure breeding plants that Mendel started off with. What are the correct genetic terms for your dogs and the two subsequent generations? Well, of course, the two dogs that you're starting with represent the P for parental generation. The puppies that would result from that original breeding would be the first filial or the F1 generation. And if you allowed two of the puppies, once they reached breeding age, to breed, then the second set of puppies that you would get would be the second filial generation or the F2 generation. So that's the answer to this question. Fairly straightforward, just checking to make sure that you are aware of the basic protocols that Mendel is setting up. So let's take a look at Mendel's first law called the law of segregation. So in the law of segregation experiments, Mendel worked with plants that differed in the traits for one character, what is referred to as a monohybrid cross. So obviously the term mono refers to one, so it's a single characteristic that represents the difference. And hybrid simply refers to the fact that it is a combination of two individuals creating something new. So this would be, for instance, a purple plant and a white plant. So he merges a white plant and a purple plant together by transferring pollen, and out of that would become would come the F1 generation, a new generation, not knowing what you're going to get out of it. Now, of course, he didn't use the term monohybrid. That's a modern terminology, but that's what he's working with. So the parents differed in the traits for a single character. And we'll just put an example here to make sure that that's clear. For example, white and purple flowers. That would be an example. And in fact, that's what we typically talk about when we're talking about Mendel's monohybrid crosses. So here are some of the characteristics that Mendel worked with. In fact, these are all 7 of the characteristics that Mendel worked with. So you've got the purple and the white for flower color, the position of the flower, so it's referred to as axial, growing off the side, versus terminal, growing off the tip, seed color, yellow versus green, seed shape or texture, round versus wrinkled, pod shape, inflated versus constricted, pod color, green versus yellow, and length of the stem, tall versus dwarf. Now, you can see here that it's also listing dominant versus recessive. Mendel actually created the terms dominant and recessive. And unfortunately, most people misunderstand what is meant by dominant and recessive. I'll explain that after we talk about the experiments for the law of segregation, because it's appropriate to talk about it there. So these are the characteristics that he's working with. These are the traits of each of the characteristics that he's working with. And he's got pure breeding plants for each of these traits, for each of these characters, because he did all 7. We know he did all 7 because we've got his data. We're lucky to have his data for that. So here's basically what he started off with. He started off with his purple flowers and his white flowers. And he transferred purple pollen to white flowers. He transferred white pollen to purple flowers. He allowed them to grow, set seed, took those seed and planted them to get his first filial generation, the F1s. And when he looked at the F1s, all of them, without exception, had purple flowers. But what was important about the purple color was that it was exactly the same purple as the purple parent. So all of these look like these purple flowers, exactly the same color. Now, Mendel had a question to ask, and the question was, what happened to the white? Where did it go? Did it get completely lost? Had it completely disappeared? Or was it hidden in the F1 generation? This was where he was smart enough to realize that if he produced the F2 generation, he could answer that question. Because if the white was completely gone, then the F2 generation should be all purple. If, however, the white was hidden, there was a chance that it would re-emerge in the F2. So he allowed the F1s to either self-fertilize or transferred pollen between the two. It really doesn't matter which way he did it, but we know he did both. He would produce the F2 generation, and sure enough, the white reappeared in the F2 generation. But it didn't just appear randomly. Once he counted up all the purple flowers and all the white flowers, he found that there were 3 purple flowers to 1 white flower. There was a very distinct ratio, a 3-to-1 ratio. Now remember, Mendel had 7 characteristics in all to look at, with these 2 either-or traits for each of the characters. And it turns out that he got similar sorts of results for all 7 characters. One of the 2 traits turned up exclusively in the F1 generation. And then in the F2 generation, 3/4 of the resulting offspring continued to show that trait that turned up in the F1, but the missing trait in the F1 now reappeared in the F2, and the ratios were always around 3-to-1. We actually have his numbers, and when you look at his numbers, and our textbook doesn't reproduce the numbers, but when you look at the numbers, only one of the characters actually gave a number that was really close to 3-to-1. All the others are close, but not exactly 3-to-1. And this is where Mendel's statistics background came in. The fact that he had a very strong math background, particularly with statistics. He knew that even though these numbers, these ratios, were not exactly 3-to-1, he could show statistically that they were or they weren't, as the case may be, by applying statistics to this. And that's what he did. So he did the statistical analysis of the data, and it turns out that while the numbers are not exactly 3-to-1 as raw data, statistically, they actually do represent a 3-to-1 ratio. And so not only did he get it with flower color, he got it with all 7 characteristics, which means this is not an accidental ratio. This is an absolute ratio, and it needs to be explained. The question is, how do you go about explaining this? And the answer lies right here, or at least the key lies right here with the F1s. So the F1 generation is the key. So why should the F1 generation be the key? Because the F1 generation is the product of 2 distinctly different flower colors. There are purple flowers and there are white flowers. And because both parents must have made a contribution to the F1 plants. I mean, this was a well understood principle that both parents make a contribution. It means that the F1s must have received genetic information for flower color from the purple parent, but must have also received white genetic information from the white parent. And from this, Mendel concluded that these individuals must have two separate pieces of genetic information or inheritance particles. One being purple, because that's all the purple flowers could pass on, and one being white, because that's all the white flowers could pass on. So these individuals must have two pieces of genetic information or two inheritance particles, a purple particle and a white particle. And they must be passing those on to the F2 generation. And somehow the way they pass on those purple and white inheritance particles is resulting in this 3-to-1 ratio. Now, Mendel did not have the modern genetics tools that we have available to us today, but early in the 20th century, a British geneticist by the name of Reginald Punnett developed something called the Punnett square. Now, I know in the Khan Academy presentation on Mendelian genetics that is part of the resource page, they talk about the idea of the Punnett square being named after a strawberry punnet because that's square in shape. No, 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 no. There was a British geneticist by the name of Reginald Punnett. He was, in fact, one of the three individuals who, quote unquote, rediscovered Mendel's work, recognized just how important Mendel's work was. And Punnett developed the Punnett square to explain what is happening every time you do a cross, or more precisely, to allow you to diagram what offspring you can expect to get from a particular genetic cross. Now, Mendel obviously didn't have access to the Punnett square. We have access to the Punnett square, and it's in looking at the Punnett square that we actually see why you get this 3-to-1 ratio from individuals who have two separate pieces of genetic information. So let me take you to the diagram where we have the Punnett square laid out. And I'm going to jump over these slides and we can come back and talk about those in a moment. So here is the individuals that represent the F1 generation. Now, a quick comment on the notation that's used here. Notice that we're using an uppercase P and a lowercase p to designate the two, as we know them today, alleles that are responsible here. The way Mendel chose to diagram the inheritance particles, or what we would call alleles today, is to choose a letter that represented the characteristic that he would ultimately refer to as dominant, and I'll tell you what that means in a moment. And so the uppercase P here stands for purple, because purple turns out to be the dominant characteristic or the dominant trait. The lowercase letter, equivalent to the letter that is chosen for the dominant characteristic, is used for or dominant trait is used for the recessive trait. And so the lowercase p is used for white here, not a W as you might think. Mendel chose to use uppercase and lowercase letters. We use a different set of symbols today because of the limitations of using uppercase and lowercase letters only. But for Mendel's work, we still use uppercase and lowercase letters, even though we have a separate system that we use elsewhere. So here are the individuals from the F1s. They are all purple because they have a copy of the purple allele that they got from their purple parent, but they also have the lowercase purple. And I, like many geneticists, where there is the possibility of confusion about whether it's an uppercase or lowercase letter, I put a bar on any lowercase letter. So the lowercase p, I usually put a bar across the tail to indicate that that's the lowercase p. So you can see where I put the bar in there. So if in fact you are using these as the inheritance particles or the alleles that you're going to pass on to the next generation, Mendel had determined that, in fact, only one of the two is ever going to be passed on to individuals in the next generation. But which one was passed on was basically a random thing. So it could be the big P that gets passed on, but it could just as legitimately be the little p that gets passed on. So with the Punnett square, we now have the ability to map that out to see what happens. So here is the big P and the little p possibilities that could be passed on. And because we're crossing two of the F1s, the other parent is going to have the same possibility, passing on the big P or the little p. Now we then just bring everything together. So these are two big P's, and that's going to give us a purple flower. If we have the big P, little p combination, well, that's the same as we saw back up here in the F1s. So that's another purple. This one is big P, little p, so that's another purple. And then if you've got two little p's, that gives you your white. And there's your 3-to-1 ratio. It does, in fact, the presence of two separate alleles, one for purple and one for white, does, in fact, allow you to explain the origin of the 3-to-1 ratio. But it also requires something else. It requires an understanding of what big P and little p, what dominant and recessive actually means. So dominant, according to Mendel, the trait that is dominant is the trait that is present in the most frequently. It's the one that is present in the greatest number and is the one that is produced if only one copy of its allele is present. So purple is the one that is present in the greatest number, and any individual that has at least one copy of the purple allele is going to be purple. In order to show the recessive trait, which in this case happens to be white, you have to have two copies of that allele. In other words, both alleles must be for the white trait. And as much as people want to say that there is a dominant allele and a recessive allele, there is no such thing. Alleles are neither dominant nor recessive. It's actually the traits that are dominant and recessive. So the purple trait is dominant over the white trait. And today we understand exactly why that is the case. The purple allele actually codes for producing purple pigment. The white allele codes for making no pigment at all. So big P, or purple, means make a purple pigment. And little p, or white, is no pigment. So for individuals that have the big P, little p combination, the big P says make purple pigment, the white says make no pigment. And so the only pigment that you're making is purple. And so you obviously are going to be a purple colored flower. And that's the reason why these individuals that are big P, little p, have the purple color. That's why the F1s are all purple. That's why 3 out of the 4 offspring in the F2s are purple, because they all have the allele that says make purple pigment. If you have the little p allele, that says make no pigment, so you're obviously making purple pigment. If you have a big P, if you're little p, well, both of those alleles say make no pigment, so you make no pigment, you are white in color. So white here is lack of color. So that's the nature of dominant versus recessive. There are other things that come out of this, and this is detailed in these notes that we jumped over. So basically, there are alternative versions of the genes. These we would call alleles. These are the alternative versions of the genes, and I introduced you to that concept in the previous presentation, but here it becomes really important now. The notion that genes come in different versions, which we refer to as alleles. So a gene gives rise to a character. An allele gives rise to a trait. That's the connection. So remember, a trait is a version of a character, just as an allele is a version of a gene. So a gene gives rise to the character. The allele gives rise to the trait. And so we have these alternative versions. Each of us then gets two alleles, one from each parent. So you have two alleles for each character that you show. And those alleles, one of them came from your mother, one allele came from your father. And you might ask, how did we get those? Well, you may recall when we talked about meiosis in the last presentation, that the chromosomes come as homologous pairs. And we have homologous pairs of chromosomes within our cells. All of our somatic cells, our body cells, have these homologous pairs of chromosomes. And one of those homologous of each pair came from your mother, one of each pair came from your father. So it stands to reason that these alleles come on these chromosomes, and the one that you get from your father came on the homologous chromosome that you got from your father. The allele that you got from your mother came on the homologous chromosome that you got from your mother. So there is an implication here of genes being located on chromosomes. And that is, in fact, one of the interpretations that people came to shortly after it was recognized in 1901 just how significant Mendel's work was, that Mendel's inheritance particles, what would become referred to as genes, must reside on chromosomes. It would take a while to demonstrate that that was in fact the case, but nonetheless, this was an interpretation. So once we recognize that there are different versions of the alleles and they're on these homologous chromosomes, and remember that is the one difference between the homologous chromosomes, while they may be same size, same shape, same location of the centromere, same banding patterns if you stain them with dyes, and the same genes in the same locations on the chromosomes, the one difference is at the allele level, because they may or may not have the same alleles on those homologous chromosomes. If they do have the same allele, then we refer to the individual as being homozygous. Remember, homo is the prefix for the same. If the alleles are different, then we say that the individual is heterozygous, two different alleles. If the alleles are different, then that's when the dominant versus recessive comes into play. So if you have a copy of the allele for the dominant trait, then that is the trait that you show, irrespective of what the other allele is. As long as you have one copy of the allele for the dominant trait, then you show the dominant trait. In order to show the recessive trait, you must be homozygous for it. So if you're homozygous for the dominant trait or you're heterozygous, you show the dominant trait. So to show the recessive trait, You have to be homozygous for it. If you're homozygous for the dominant trait or you're heterozygous, then you're going to show the dominant trait. So you can see here that the notation, and this is, and I followed the notation in the textbook, it talks about dominant alleles and recessive alleles. Just want to remind you again that alleles are neither dominant nor recessive. It's actually the trait that is dominant or recessive, not the alleles themselves. Cells do not differentiate between dominant alleles and recessive alleles, or alleles for the dominant trait and alleles for the recessive trait, to use the right terminology. If one of the two alleles is on, then the other one is on as well, and both are active at the same time. It's really the way the traits interact with each other that determines the final physical appearance. So in the case of purple and white, here in Mendel's peas, remember again that the dominant trait is producing purple pigment. The recessive trait is producing no pigment at all. So if you are heterozygous or homozygous for purple, then you're making the purple pigment irrespective of what the other allele says, so you're always going to look purple. If you're homozygous for the recessive trait, then both alleles are saying make no pigment, and so you look white under those circumstances. So it's really all about the product of the allele, the actual trait itself, that determines the nature of dominant versus recessive. And that will be reinforced later in this chapter as we dig deeper into some other ideas that will fall a little bit outside of Mendel's observations. And so the notion of the law of segregation really traces its origin back to what goes on during meiosis, during reproduction. Because the law of segregation is basically stating that during the reproductive process, and we could say during meiosis, the two alleles get separated or segregated from each other and wind up in different gametes. So one of the two alleles will wind up by itself in gametes, and the other allele will wind up by itself in other gametes. So there will be gametes that, in the case of flower color, there will be gametes that have the purple allele, and there'll be other gametes that have the white allele. That's the whole basis behind Punnett's Punnett square, as we saw earlier. That separation or that segregation, that is simply the basis for Punnett's Punnett square. When the egg and the sperm unite at fertilization to create the zygote again, you bring back together the pairs of alleles, and it is the interaction between the traits produced by those alleles that give rise to the particular characteristics or the particular traits that you're going to see. Now, we need to introduce a couple more terms. They're actually used on this diagram, but they're not defined yet. So the genetic makeup is called the genotype. The physical appearance is called the phenotype. So when we start looking at the results of the law of segregation, there are two ratios that we need to actually look at, and those are documented down here. So if we're looking at the physical appearance, we know that it's 3 to 1, 3 dominant trait to 1 recessive trait. And so when we're talking about traits and physical appearance, then we are talking about phenotype. So the phenotype ratio is the 3 to 1 ratio. And that's true for any Mendelian monohybrid characteristic. If we're talking about monohybrids, and we're talking about something that is inherited in a Mendelian fashion, then in the F2s, the ratio is always going to be 3:1 for the phenotypes. The genotype ratio, however, is 1:2:1. One homozygous dominant, and that's the correct way of referring to this individual. That's how we genetically refer to this individual. Homozygous dominant, 2 heterozygous individuals, and 1 homozygous recessive. So there are, in fact, 3 different genotypes: a homozygous dominant, a homozygous recessive, and a heterozygous, and the ratio is 1:2:1. So there are 2 ratios that you need to remember, and in fact, you can remember these because these are absolutes. The phenotype ratio is always going to be 3:1, the physical appearance ratio. The genetic makeup ratio or the genotype ratio is always going to be 1:2:1. These are absolutes. So long as you remember that, you can't go wrong in terms of understanding what you're looking at. So if you're given some numbers and you're given some numbers based on physical appearance and the ratio comes out at 3:1, you know you're looking at something that is Mendel based. If you're given some genotype ratio data and the ratio comes out at 1:2:1, again, you know you're dealing with something that is Mendel based. These are absolute. So we talked about the Punnett square and where that came from. We talked about genotype and phenotype. They're only just introducing these now after they've already put them into the diagrams in the book. And I'm following the flow in the book. So I prefer to introduce them earlier, but this is the flow in the book, unfortunately. So I put the terms in where I put them in. So I've already mentioned the notion of these being located on the homologous chromosomes. This diagram just simply illustrates that for you. So here you can see the homologous chromosomes for a particular individual. Here is a gene where the individual is homozygous dominant. So homozygous for the dominant trait. Here is in this location, they're homozygous for the recessive trait, lowercase letters. Here, they are heterozygous. So this is how we trace these back. And remember, what is going to happen to these chromosomes during meiosis? Well, if they're homologous chromosomes, members of the homologous pair, we know that they're going to get separated. So it's the fact that these get separated during meiosis that leads to the segregation. And we should remember where they're separated. This will be during anaphase. And it's going to be anaphase 1 when the homologous chromosomes are separated. So this is where the law of segregation comes from. The fact that the homologous chromosomes get separated during anaphase 1. So let's take a quick break again. And here are the concept check questions, which I will answer when we come back from the break. So let's take a look at these. If 2 organisms have the same genotype, must they have the same phenotype? If they have the same phenotype, must they have the same genotype? Interesting question. Mendel showed that all organisms have 2 alleles for every trait. Where do these alleles come from? I'll see you on the other side of the break with the answers.